the idea behind how it was possible back then requires you to understand a few few separate techniques first, and when you put them all together, you have this really ingenious way of really profiling someone based on their DNA. And I'm going to like split it into a few parts. And part one is a technique that allows us to chop up DNA molecules based on their sequence. Okay, and this last part of the sentence is kind of important, and what I mean by based on their sequence is something like this. So say I have a specific sequence of DNA. Okay, I have this sequence of DNA. I'm just going to call it sequence A, and what I'm going to do is I'm going to treat this with some chemical. Okay, let's just call it chemical X for now. So I'm going to treat this sequence of DNA with this chemical X, and what chemical X is capable of doing is it's going to make cuts on the DNA. Okay, it might make a cut here, it might make a cut here, it might make a cut here, it might make a cut here. Okay, and what's special about this chemical is that it's not just making cuts entirely at random. It will always make the same cuts exactly at the same places for the same sequence. So if you treat the same sequence, I'm calling it sequence A again to indicate it's the same exact sequence, then it'll make the cuts at exactly the same spots. See, it's going to cut the DNA exactly the same way. On the other hand, if I had treated it with a slightly different sequence, or maybe a very different sequence, so say I have a completely different sequence and I'm going to use a different color to indicate that, and it might even be of a different size, and the idea is treating this this new sequence with our mysterious chemical is going to give completely different pattern of cutting. So you're going to get a cut here, you're going to get a cut here, you're going to get a cut maybe here, maybe all the way here, maybe here, right? And it's important to know to understand what we mean by cutting it. At the end of all these cuts, what we're essentially going to get is we are going to get a set of fragments, right? So sequence A is going to have fragments like these. So there's going to be this fragment before the first cut, which is, let's say, about this big. There's one after that which is slightly bigger, then there's one which is smaller, and then there's one which is about the same size, and then there's one that's really small, one that's kind of big. Let me draw it a bit better, and there's one that's here, right? And this you could maybe do a better job of sorting these by size, but I've just drawn it as you see it from left to right, right? And you're going to get this size pattern of cuts every time you treat the same sequence A with the same chemical. Whereas if you used a different sequence, that is sequence B, what you seem to get is something like this. You get like a long piece, that's this one, then you get a slightly shorter one, then you get a slightly longer one, then you get one that's about the same size as that one, then you get another long one, and so on, right? And the idea is that what you seem to have is two different size profiles for the fragments. You might end up with a different number of fragments itself, and they're all different sizes. So okay, that's basically just a technique that can chop up DNA in a specific manner, and we'll get to see why this is important. But for now, let's try and put a name to this. So this blue chemical, this thing is something that's called as a restriction enzyme. And what it is it's basically just a protein, and what it does is when it comes in contact with a specific sequence of DNA, and typically it's a sequence that's very small. So maybe this is something like GACTC or something like that. So every time it comes across a GACTC, it's going to make it easier for the DNA to split apart at that point. So it's going to catalyze a reaction that causes the DNA to basically cleave at that point. So and the reason why it cleaves the same sequence in the same way is because the same sequence is going to have the same GACTC or whatever the sequence it likes at exactly the same locations, whereas a different sequence is going to have that sequence at different locations, right? So this whole technique as a whole is called, there's a name for the technique itself, and it is called restriction digestion. Okay.